Hello.
Hi, wow, my headphones were really loud.
I think I might have just been really loud too.
No, my headphones were set at like 60, which is not great for my in-the-ear headphones. Hi, everyone. If my glasses are too distracting, I'll put on different ones because I know they're like giant and catch the glare of any light around me.
Look fine to me.
Riley said we love the glasses. Um,
Hi, Tim. Hi, David.
Hello, thanks for inviting us.
Hello. Hi, Diane. How is San Diego?
Gorgeous. What else is new?
I shouldn't have asked.
I swam. It was beautiful swimming this morning. So yeah, it's
How's the water?
well, we did. I must say, we did have a little bit of marine layer, so it was chilly out. So it was like 60.
We had snow.
I know. I heard.
Where did we have snow? Where was it?
Kalkaska had about 8 inches up north.
We just had some flurries around my place out at the bus stop this morning.
Doug is supposedly here somewhere, making noise.
Yeah, I
There
can
you
hear
are.
you. You guys able to hear me?
Hear you. Your screen's just black. Oops, wrong button. You think since I use Google Slides for everything, I would like know how to use it, but I don't. Hey Lars, are you able to see when people come into the waiting room?
Yeah, it had been letting me know
Okay.
before.
Can you, while I'm presenting,
Yep.
can you just like let people in?
Oh, I just tried and somebody beat me to it.
It was me. I beat you to it.
I can keep an eye on it, sure. I got that one.
Okay, so
I have some workmen at my house making noise outdoors, so I'm gonna try to be on mute.
I also have that, and my apartment complex did not warn me. So if you suddenly hear a nail gun, it is that. They are right outside my window. So hopefully this goes fine. This gets me to what I was about to say. Just for like the ease of the Zoom, can everyone who's not currently on mute, please mute themselves? Also, I love that people are putting things in the chat. I cannot see it. I just see the alert that a chat has been done. So I'll check the chat at the end, if that's okay with everybody. Cool. So it's 2:01. So I'm going to begin since we don't have a lot of time and I have a lot of ground to cover.
We might want to hang on a little bit, actually. I'm not sure that we have Jessica yet.
Is she okay? She said she was going to come, right?
Mm-hmm. So I think maybe we'll just wait another minute. I'll send an email if we don't see her in another minute or so.
Allison Zahoric is being so nice. She just keeps messaging me, you're going to do great. Oh, that Jessica. Yes. Admit. Oh, you got me. You beat me to it. Now can I start?
I think so. Let me just double check. Jessica, do we have you here, available to hear and stuff?
Yeah, I'm just trying to get my Windows set up. For some reason, it's stuck in my wrong monitor.
Sure, sure.
Want a second?
No, it won't let me move it.
All right. I can just maybe start by saying welcome, everybody. Thanks a lot for joining us. So I'm Lars Brudvig. I am Toby's advisor. And as you see on the screen, Toby's other committee members are Jessica Meisel from Plant, Soil, and Microbial Sciences, David Rostein from Forestry, and Doug Landis from Entomology. And this is Toby's proposal defense. They joined our group in, let's see, it was fall 2019, and has established really fantastic and interesting research about the carbon consequences of reintroducing fire to long fire suppressed oak savannas, which is based around an experiment that Doug Landis and I set up over 10 years ago. You'll hear, of course, all about what Toby's been up to and what they're planning for the future. But I'll just say the research has brought such an interesting new angle to the project. So I'm really glad that Toby's gotten involved and has brought so much through their research. So with that, I can hand things over to Toby. I'll keep an eye on the chat in case anything important pops up. I will also keep an eye on the waiting room so I can let people in.
Cool. Thanks, Lars, for the generous introduction. Hi, everyone. Hopefully you can hear me okay. I'm losing my voice a little bit, but I'm going to try to go strong through this presentation, so just bear with me. So the title of my talk is Fires, Earthworms, and Restoration Are There Hidden Carbon Costs to Restoring Fire to Degraded Savannas? So I'm going to start out with my land acknowledgement. So Michigan State University occupies the ancestral, traditional, and contemporary lands of the Anishinaabe, Three Fires Confederacy of Ojibwe, Odawa, and Potawatomi peoples. In particular, the university resides on land ceded in the 1819 Treaty of Saginaw. We recognize Michigan's 12 federally recognized Native nations, historic Indigenous communities in Michigan, Indigenous individuals and communities who live here now, and those who were forcibly removed from their homelands. In offering this land acknowledgement, we affirm Indigenous sovereignty, history, and experiences. So my outline for today is pretty typical for any proposal defense in PLD. So I'm going to give you some background intro. What am I studying? Some brief information about restoration of grassy biomes. We're going to talk a little bit about fire, and then I'm going to talk a little bit also about the carbon cycle. I'm going to go over my proposed questions, of which there are 4, and then we're going to go through chapter by chapter for the 3 chapters in my proposed thesis. So starting with my introduction. So humans have altered ecosystems globally through the conversion of land for human uses, especially through agriculture, urban development, and mining. We've also altered ecosystems by changing the disturbance regimes that ecosystems normally experience. So humans are huge fans of stability. We often exclude disturbances, especially fire, from the places that we live. And that sounds great, right? We all grew up with these kinds of adverts about how only you can stop forest fires. So isn't stopping disturbances like fire kind of what conservation and ecosystem health is all about? Well, not really. So as it turns out, global burned area has decreased nearly 25% in the last 20 years, with 90% of this decrease occurring in grass-dominated systems. And that's actually pretty bad for these ecosystems because grassy biomes like prairies, savannas, and shrublands actually thrive with a disturbance regime. They've evolved over millennia to be fire adapted. So many of these organisms that live in grassy biomes, especially the plants, have specialized resprouting systems and specialized storage organs to help them deal with constant fire. So when you exclude fire from these systems, they actually undergo environmental degradation. So this global decrease in burned area has caused grassy biomes to undergo degradation on a global scale, converting many grasslands, prairies, and savannas into shrubby woodlands. And so this conversion from, previously open canopy grasslands to kind of more closed canopy woodlands and forests is likely globally consequential for the carbon cycle. And research is required to determine what impacts this kind of broad woodland conversion may have on the carbon cycle. Over here on the right is a really famous example of how just management decisions on the exclusion of fire can have a huge difference in what an ecosystem looks like. So this is from Van Alk in 2009, which is one of the first big papers to talk about broad woodland conversion and the reasons for it. So it's an aerial photograph of a fence showing the effective management differences with higher woody plant density and cover on the right and lower woody plant density and cover on the left. And the only difference between these two ranches shown in the photo is that the ranch on the right completely excludes fire, while the ranch on the left has prescribed burns. So you can see that removing fire is pretty consequential for how these ecosystems develop and maintain themselves. And I'm really interested in grass-dominated systems, especially prairies, grasslands, and savannas, because these ecosystems account for 40% of the terrestrial land cover. They account for 30% of global primary production, and their carbon balance drives the interannual variability of the global carbon sink. So degradation of these systems can lead through increased climate warming through higher CO2 emissions as the vegetation changes from these fire-adapted forbs and grasses to fire-sensitive trees, or as we convert these ecosystems from these long-rooted organisms to these short-rooted crop plants. So on the right is this really famous image of the difference between what prairie plant roots look like versus what agriculture looks like. So you can see the man standing here. He is actually too short. So the roots of the prairie plants are longer than his entire body, and he is 6 foot tall. But the agricultural plants right next to him are very short rooted. So the way that I like to think and explain grassy biomes to people, because a lot of people don't understand that they do store a lot of carbon, is that everything that you see in forests is pretty self-explanatory when it comes to carbon storage. All that carbon is above ground in the body of the tree. It's kind of the opposite for grassy biomes, where everything that stores carbon is underground. So something that Joe Veldman and a lot of his contemporaries like to say is that basically grassy biomes are underground forests in these extensive root systems and tubers that store carbon below ground in these grassy biomes. So because of their size and importance, I think it's critical when we think about restoring the former structure and biodiversity of these grassy biomes, that we think critically about what it means to actually reintroduce fire into these systems, since so many of them have had fire removed from their daily disturbance regimes. So we have this recognition that altered fire regimes and ecosystem shifts that I've been talking about could be consequential for global carbon dynamics, but we don't really have a critical understanding of how fire suppression followed by fire reintroduction affects specific ecosystem processes. Fire causes drastic changes to ecosystem structure via both direct and indirect effects on plant, bacterial, and animal communities. And this can alter how nutrients are stored and cycled. I focus a lot on decomposition. So decomposition here is the breakdown of organic matter to its component organic and inorganic parts, which recycles key nutrients, compounds, and elements through the finite space of the biosphere. And I personally think decomposition is a critical process to study in the context of climate warming, because it provides the link between these terrestrial and atmospheric carbon pools. But however, we don't know how the return of fire to these grassy biomes, especially after they've become degraded, affects decomposition, even though we're using fire as a restoration tool in most of these restorations. So I want to look at how fire shapes the plant community, and in turn, how that shapes the way different decomposer guilds between invertebrates, earthworms, and the microbial community, how they contribute to the decomposition of plant litter. So how is the ecosystem recycling itself is what I'm trying to ask here, and who's doing that recycling? We already know from the literature that plant identity is a dominant driver of decomposition worldwide. So it really matters what you're decomposing. But then we also know that fire itself can fundamentally alter the structure of the plant community by promoting the growth of fire tolerant or fire adapted species while reducing the number of fire sensitive species. And then we also know invertebrate decomposers and microbial communities within an ecosystem contribute differentially to decomposition depending on how much litter is available in the system, what that litter quality actually is. So what are the carbon, nitrogen, and phosphorus ratios in that litter? Is it high quality litter or low quality litter? Because that changes what each community is willing to decompose. And then we also know that invertebrate decomposers and microbial communities respond differently to fire as well. So when we're talking about leaf litter and the quality of leaf litter, fire can lead to a change in these carbon, nitrogen, phosphorus ratios of the litter post-burn, and the subsequent nutrient limitation can actually decrease microbial mediated decomposition because it's harder for microbes to decompose this lower quality litter. But fire is also interesting because it can cause nutrient pulses as it's essentially acting as its own consumer of this leaf litter. So it's breaking down these leaves into different nutrients, but it can also cause biochar formation. And both of those things have different consequences for ecosystem nutrient cycling, especially carbon cycling. So if you cause a nutrient pulse, you could cause an increase in microbially mediated decomposition, but you could also cause a decrease if the resulting litter quality is too low. So it's really context dependent. Ooh, suddenly my slides won't advance. Oops. There we go. So something that's really interesting to me from the Butler 2019 paper is that fire can actually increase the contribution invertebrate decomposers have to decomposition. So invertebrate decomposers like springtails and millipedes are really mobile, right? So they can move across the landscape and consume more leaf litter that's of lower quality to meet their energy demands. And this might point to some sort of relationship between these macroinvertebrates and similar decomposers and the microbial community. But there's something extra spicy over here in Michigan when it comes to studying decomposition and actually within the continental US itself. So you've probably all seen the Atlantic article by now that's talking about how the continental US is actually undergoing an invasion of non-native earthworms from Europe and Asia, especially the Japanese jumping worm. And the thing about earthworms is that they can interact with kind of every trophic level within a soil food web. They're ecosystem engineers. So these worms and their invasion are fundamentally altering decomposition in most ecosystems through some sort of feedback on invertebrate and microbial decomposers. So a couple of examples of how earthworms can affect an ecosystem is that invasive earthworms can actually increase decomposition rates because they're eating up all this organic matter and emitting carbon dioxide, as well as stimulating microbial decomposition by making complex C compounds into simpler C compounds, making them more accessible. So they're basically making food for these microbes. That's so nice of them. However, worms are also interesting because they can actually also decrease decomposition rate in a certain way because they can increase the stabilization of organic matter after they've consumed it, since these smaller C compounds and particles that they produce after they've eaten the organic matter can actually be attached to soil aggregates and become protected from decomposition. So as a novel decomposer to an ecosystem, it's currently unknown how this invasion of earthworms affects other decomposers in the ecosystem and what the cumulative effects of this invasion are on the feedbacks already known between decomposition and carbon flux from soils. And it's also unknown if invasive earthworms can be controlled, especially when we use fire as a management tool as we're trying to restore these grassy systems. So all of this information kind of led me to a question, which is, well, what if there are hidden carbon costs to restoring grassy biomes using fire? Fire already has the explicit cost because you're literally setting carbon on fire in the form of the plants being consumed by the burn. But what if there are other hidden costs because fire is affecting the invertebrate or the microbial community in some way, or if it's fundamentally changing the relationships between these invertebrate decomposer guilds and the microbial decomposers? And what if this is especially exacerbated under this earthworm invasion, since the earthworms themselves can act as another novel disturbance in the ecosystems? So I have 4 proposed questions kind of around this. So who are the current communities of decomposers who are decomposing leaf and plant material in this restored oak savanna that I work in? Under this reintroduction of fire, what are the relative contributions of microbes, invasive earthworms, and native invertebrates to decomposition? By what mechanism does fire influence leaf decomposition? And does the invasion of non-native earthworms alter decomposition rates via some direct effect, like their consumption of leaf litter, or is it through some indirect effect by influencing the relationships between decomposer guilds? And can their invasion be controlled by prescribed burns? I know I sound a lot like this when I talk about it, but I promise I have this all laid out in a much simpler way than what we talked about before. So here is actually the kind of model that I'm thinking of when I'm talking about the effects fire can have on these different decomposer guilds. So here's my big figure. So here you have fire at the top. The red lines are going to be direct, or the solid lines are direct effects, and then the dashed lines are indirect effects, and then the red is signifying a negative relationship, and then the blue is signifying a positive relationship. So some of the things I'm talking about is fire can have these indirect and direct negative effects on the plant community, on invertebrate decomposers, microbial decomposers, and possibly on these invasive earthworms. But that in turn has effects on how each of these decomposer guilds affect each other. So one of the examples here is that palatable plants can increase invertebrate decomposer populations and thus decomposition, but an increase in unpalatable plants post-burn will reduce decomposer populations or what they're willing to decompose, and thus reduce decomposition, which will lower the decomposition of plant matter overall. And then the invasive earthworm example here on the right is kind of the same deal as what we were talking about earlier, where we know invasive earthworms can increase decomposition by their consumption of organic matter and increase CO2 emission, as well as stimulating microbial decomposition by making C compounds more accessible. But if that's outweighed by their stabilization of organic matter effect, then it's kind of up in the air how we expect that to affect plant decomposition as well. But I would expect it to be lowered, since that's what it seems to be like in the literature, regardless of how much organic matter they seem to be consuming and CO2 they emit within these different microcosm studies. But we don't know because it's context dependent. So my context is going to be McCready Reserve. So like Lars mentioned earlier, McCready is a site that we've been using, or his lab's been using for the last 10 years. So McCready Reserve is actually an amazing place to do research. It's a mixed savanna woodland that's managed by Michigan State, and it's open to the public. You could go out for a hike right now if you wanted. But McCready already has these established plots and transects within the reserve that have gone restoration via fire or that have not been managed. So 5 around 1 hectare fire suppressed oak savannas were randomly assigned a fire reintroduction treatment in 2010. And then unburned plots were assigned the following year. There's 5 unburned plots as well. And burn treatments have been burned every 2 to 3 years since 2011. So we have done these prescribed burns throughout the last decade. I helped with a prescribed burn just this spring. And so when it comes to my study site, you can see right here on the left an aerial photo of McCready. Purple is the public trail system, and then the yellow are the outlines of each plot. The initials stand for what treatments the plots are in. So UN is for unburned, BO stands for burned only, and then TB stands for thinned and burned plots. Please note that for my thesis, I don't look at the thin and burn plots. I'm only looking at the burn only plots and the unburned plots. And then on the right is a photo Lars took when we set up my plots last year, where you can see my transect as well as the pitfall traps that I used for my experiments. So I have 3 proposed chapters that try and tackle the 4 questions that I talked about earlier. So chapter 1 is going to be my site characterization, where I also talk about, or where I also investigate, what the current invertebrate community is down at McCready, and how it's different between our burned and unburned plots, as well as trying to figure out who are the invasive earthworms in this system. Chapter 2 is a series of decomposition experiments that aim specifically at finding what mechanisms by which fire is leading to a change in decomposition rate. And then chapter 3 are earthworm exclusions and effects on decomposition, where I'm trying to figure out the specific effects earthworms are having on decomposition in this system, and if that's different between plots that have been burned versus plots that have been unburned. So for chapter 1, I'm going to work to answer question 1, literally who is out here doing this work of decomposition. So who's decomposing all this leaf and plant material down in this restored oak savanna? So I think this is important to find out because it's these invertebrate guilds who perform decomposition and also the ones who are interacting with these invasive earthworms that are now pretty established at McCready. So characterization of who is actually out here doing the work is necessary because understanding the contributions, of different decomposer groups to decomposition requires knowing both the effects of the groups on one another, but also their identity. So for chapter 1, I decided to use pitfall traps. So pitfall traps are a well-established method for insect sampling. So what I did is I have pitfall traps with plastic roofs to prevent flooding installed into my 10 plots at McCready Reserve along 2 10-meter transects within each plot. I use propylene glycol as my preservative solution, which kills the insects as they fall into the cup. And then these pitfall traps were sampled 6 times over the course of the summer growing season to capture the initial community, what peak community looks like in the middle of the growing season. And then at the end, as we wind down towards fall, how does that invert community kind of wind down? And then I did this for 2 summers, 1 summer during a burn year and 1 year where no burning occurred in all of the plots. So here's what my pitfall trap looked like. This is before it's activated. So you can see that my pitfall trap has this little plastic roof. It's held together by landscape staples. And then you can see that there's a 16-ounce cup buried in the ground. So this is actually a double wall design where the first cup does not leave the ground at all. So it keeps the soil stable. And then the second cup within that cup is actually the collection cup filled with propylene glycol and water to kill the organisms. And then obviously when I do my actual sampling, the cup is not covered like this by the little plastic top. And the roof is supposed to prevent flooding, but it also helps prevent interference by animals because something that happens a lot when you're pitfall trapping is raccoons are like, great, free lunch. And they'll actually eat the organisms out of your cup. So that's the other reason I put a roof on them. The earthworm sampling has to be a little bit different because of earthworms being kind of more in the soil and soil surface. So for the earthworm sampling, I decided to use the same transects as my invertebrate surveys, but instead of trying to dig them out of the ground or something like that, I actually used the hot mustard extraction method. So the hot mustard extraction method is basically you take a 1/4 meter squared quadrat of PVC or what have you, and you place it on the forest floor and you remove all the litter around it. And then you use mustard solution, which is literally mustard flour and water. And you mix it up and pour it on the soil. And the chemical within mustard flour that makes it spicy, it makes it good for us to make food like barbecue and things like that, is actually really irritating to earthworms' skin. So earthworms breathe through their skin, and so they're really sensitive to stuff falling onto their skin. And so the hot mustard actually irritates their skin so much that they start literally hurling themselves out of the ground to the soil surface to avoid the hot mustard solution. So I would pour a gallon of mustard solution out onto each of these little sampling quadrats and collect the earthworms that came out. And in case it doesn't seem clear or you've never been in McCready before and seen my plots, this is actually what my transects looks like. So Lars does a yearly vegetation survey along this vegetation transect. Nothing happens within that vegetation transect for me because I don't want to interrupt any of Lars's sampling. So what I decided to do is I decided to choose the slope midpoint. And the reason I chose the slope midpoint to make my transects is because McCready is actually really hilly. Because of the history of glaciation in Michigan, a lot of the plots at McCready are actually set on a hill. So the transect for the vegetation transect starts at 0, which is the bottom of the hill that are usually swamps. And then the top of the transect is actually the top of most of the hills at McCready. And that is actually looks more like a forest or a savanna, depending on which plot you are in. And so for my vegetation for Lars's vegetation transect, I chose the slope midpoint, which for most plots is around 30 or 40 meters. And then at the outside edge of each of the vegetation transects, I drew my own 10-meter transect. So the invertebrate sampling, so where I install my pitfall traps, happens at the 2, 4, 6, and 8 meter marks on my midpoint transect, while the earthworm sampling happens at 3, 5, 7, and 9 on the midpoint transect for most of my plots. These are not right next to each other. They're kind of offset from each other. because I don't want any of the mustard solution sampling, so any of that solution, since it does run downhill, I don't want any of that to affect what could be falling in my pitfall traps, because it's also irritating to some other organisms like ants and beetles. So I actually tried to do the earthworm plots a little offset from where I have my pitfall trap sampling. But this is what it looks like. And then when it comes to what I'm going to actually use all this data for, so I'm basically just going to do a bunch of t-tests. So mostly I'm going to try and compare the total number of invertebrates between burned and unburned plots that I've captured in my pitfall traps. And then I'm also going to compare species richness between the plots that have had fire reintroduced to them versus the ones that have been completely unburned. And then I'll also use a permanova to analyze the difference in invertebrate communities between the burned and unburned plots and visualize those using an NMDS plot. So how similar are these communities to each other versus how dissimilar they are. And then for the earthworm stuff, I'm going to do kind of the same thing where it's just like a bunch of t-tests to compare the number of unique species in each treatment. I'm pretty sure I only have Lumbricus terrestris actually when it comes to these earthworms, so this will probably change. But I haven't gotten through the analyses of the earthworms I've caught for this year, so that could change. And so the other thing, if there's only one species of earthworm present at each treatment type, then I'll just simplify this into comparing the capture rate between plots. So am I catching more earthworms in burned plots or unburned plots? And then the other thing I did for my first chapter in characterizing my site is a carbon budget study. So this involves placing 4 1 meter by 1 meter subplots in my transect and clipping all the biomass down to that dust layer or soil surface and sorting it into live or dead, woody or herbaceous matter. I've also characterized what the fallen wood and logs in these subplots look like in terms of their decay state, how big they are. And then I use allometric equations to calculate how much biomass and biomass stored as carbon is in these fallen wood logs. And then because I also want to know details about the soil, like the soil carbon and soil nitrogen, I've also taken 4 mineral soil cores at each of these 1 by 1 meter subplots. And I've also sent them out to see their soil CN ratios, their overall C content and soil moisture. And then because there's the ability to capture obviously trees or shrubs within these subplots that are pretty big, I didn't actually cut down things that were bigger than 5 centimeters around. I just measured them to the nearest centimeter or 0.1 centimeters, and then also used allometric equations from Griegel and Ullman to calculate their biomass and carbon with these allometric equations. And then what I do with these clip plot biomasses that have been sorted into these discrete categories is that I've dried and weighed them, and I'm using their masses to estimate the C in each of these components. Again, using these allometric equations. So basically I'm cutting a bunch of stuff down, drawing it and weighing it, and then doing a lot of math. And then the other thing I did to characterize the soil is I also took shallow soil cores, specifically along the length of the earthworm surveying transects, to see if there's a difference in the soils between where I've done these clip plots versus where I'm doing my earthworm sampling, since the soil at McCready is pretty heterogeneous. And then when it comes to this carbon budget stuff, it's still going to be a lot of t-tests comparing how much mass is in these in all of the live or herbaceous, live or dead, woody or herbaceous categories for these clip plots between the burned and unburned plots. And I'm especially interested in seeing if leaf litter mass, dead woody biomass, and woody biomass is different between these two treatments, between burned and unburned plots, because you would hope that you would see less leaf litter and less woody biomass hanging around the burned plots, since the fire should theoretically take care of those. But we'll see. I am not done processing all of the clip plots yet. I have 1 bag left to dry and weigh, and then I have to process a lot of the leaf fall as well. And in case you are confused about where these plots are in relation to my transect, so up here you can see that I have my slope midpoint transects up at the top. And then because I wanted to get a good estimate of what the clip plot should look like. So the clip plot should mirror what overall McCready sites look like. I decided to do 4 of them. And so there's one on each side of the vegetation transect at the end of a 10-meter transect. So that's what these purple squares are. And so at the end of, at 10 meters and at 20 meters, at the end of a 10-meter transect, I've put down the clip plots. So there's one on the right side, there's 2 on the right side of each plot, one at 10 and one at 20. And then on the left side of the plot, there's also one at 20 and one at 10 to hopefully get a good sampling of overall what the vegetation community looks like at McCready. I didn't want to do it on my invertebrate sampling transect because one, these clip plots are really big and I didn't want to disturb the soil or the leaf litter that is around my insects because it could affect what could fall into my cups or what I could capture in terms of earthworms. So thankfully, the last 2 summers, all I've been doing is kind of this invertebrate and earthworm sampling as well as these clip plots. And as you can see here on the last day of collecting last summer, I am very grateful that this is done. It is so hard to carry 4 gallons of mustard solution across a swamp, which is where some of my plots are located at McCready. So this is all done. I just need to process the data. But chapter 2 is the stuff I'm going to be working on in the next year to 2 years. So chapter 2 is all about clarifying which mechanisms, if any, account for the differences in decomposition between these burned and unburned plots. So I'm going to do this sequentially, working from changes that can arise due to differences in relative contributions of decomposer guilds to decomposition to the specific effects that plant species have on either plant identity or palatability. So I'm going to do this tracking decomposition using litter bags. So litter bags are a really common tool used to track decomposition. So it's basically tracking the mass lost from when you first put the litter bag down versus when you collect it sometime later. So I'm going to use litter bags that are 12 by 6 centimeters because that's the most common size used in the literature. And I'm mainly investigating 3 different mechanisms that could mediate these differences in decomposition between plots. Sorry, 1 second. Thank you. So for this one, going back to my big figure, what I'm looking at here is how the plant community is affecting either these invertebrate decomposers, which is circled here, and how that in turn leads to some change in the decomposition of plant matter. So if you've never seen a litter bag before, here's a really good example from the LIDAT studies at Oregon State University over at the right. And the first mechanism that I'm going to look at is reintroduction of fire to the savanna changes the relative contribution of different decomposer guilds to decomposition. So I'm going to be using nylon mesh, and what these litter bags are going to look like is I'm going to take the dominant overstory plant species in burn plots or unburned plots, so Quercus alba or Acer rubra respectively, and I'm going to recollect a subset of the litter bags every 3 months throughout the year to get some sort of decomposition rate from each of the sites. So I already have all this leaf litter, so I set up litter fall traps thanks to Dr. Meisel, and I recollected litter each week from these litter fall traps for each week in the fall during 2020, and that's the litter that I've included in the decomposition bag. So at each site, I'm going to deploy these litter bags, and I'm going to have 2 different kind of flavors of decomposition bags. So I'm going to have some litter bags made from small bore mesh, which has really small, like no-see-in mesh size holes. And this really small mesh excludes invertebrates. So invertebrates cannot get in it to munch on this leaf litter. And what these small bore mesh bags do is they track microbially mediated decomposition. And then I'm also going to have a set of large bore mesh bags that have really big hole sizes that allow invertebrates access to leaf litter, and this will allow invertebrates to get in there and also perform decomposition. And then because I have bags that track microbially mediated decomposition, and then some that allow both microbes and invertebrates to come in and chow down on this leaf litter, I can use both of those bags to calculate what the contribution of just invertebrates is versus the contribution of what microbes are. So I can have both of these rates of decomposition. So I'm getting their contribution that way. So the mass loss from these big litter bags will capture both microbial and invertebrate mediated decomposition. And since I have those microbe only decomposition rates from the small bore mesh bags, I can calculate the relative contributions of the different decomposer communities to decomposition and identify if these contributions differ between burned and unburned sites, since I'm deploying the bags in both areas. And then for the stats analysis here, I'm going to be doing a split plot ANOVA where kind of the big plot is the fire treatment, so burned versus unburned. And then the plots nested inside that big plot are the mesh size treatments. So is it a small bore litter bag or a large bore litter bag? And then decomposition rate is the dependent variable. So once I do that, I'm going to move on to mechanism 2, which is looking at how plant identity is going to change decomposition rates. So mechanism 2 is reintroduction of fire forces changes in plant community structure, which makes litter less palatable to decomposers, which slows decomposition. So I'm basically going to do the same thing as 1, where I'll create a set of litter bags with the same distribution of small bore and large bore bags. And then these litter bags are going to evaluate plant identity effects on decomposition. So they're going to look at the consequence of prescribed burn shifting tree composition in favor of oaks over these maples that are more common in unburned plots at McCready. So I will create litter bags with either red maple or white oak leaves and then reciprocally transplant those bags into burned or unburned plots. So litter bags made from maple leaves will be placed in burned plots, and oak leaf litter bags will be placed in unburned plots. And just like last time, the statistical analysis here is going to be a split plot ANOVA with 2 factors, where burn treatment is the big plot, and then mesh size and litter species are our 2 factors. And just like in the first mechanism, the dependent variable is still going to be decomposition rate. And then mechanism 3 is kind of in the same flavor, but instead of changing which plant species is going into each litter bag, it's just changing the origin of where that leaf litter came from. So mechanism 3 is going to be reintroduction of fire causes changes within plants themselves, which render litter less palatable, slowing decomposition by microbes while hastening it for mobile decomposers. So for this one, I'm also going to create litter bags with the same distribution of small and large bore mesh as in mechanisms 1 and 2. But what's going inside the litter bags are just going to be Quercus alba, which is the dominant overstory plant in both burned and unburned plots. And this litter is going to be analyzed beforehand for carbon, nitrogen, and phosphorus ratios to ensure that there's some sort of difference between burned and unburned plots, because one of the big hypotheses that came out of the Butler et al. Paper 2019 was that even if the plant community itself doesn't shift in response to fire, plants themselves start creating lower quality leaves when they are repeatedly burned. So I'm going to check for these carbon, nitrogen, and phosphorus ratio differences, which are indicative of leaf quality. Before I start this experiment, to make sure there's actually some sort of difference, like is this a real effect that the Butler et al. Paper found between these burned and unburned plots? And if I do find a difference, so like, yes, there is lower quality leaf litter in burned plots versus unburned plots, I'm going to go ahead and make my litter bag. And then just like before, it's the same small or large bore mesh as in the first 2 litter bag experiments. But I'm going to do a little bit different. So the litter bags made with the Quercus alba litter from burned plots will be placed into unburned plots, and vice versa for the unburned plot litter. So litter that I've collected from unburned plots is going to be placed in litter bags and put into burned plots. So this reciprocal transplant will evaluate how fire-mediated changes in leaf litter carbon, nitrogen, and phosphorus ratios affect the contribution of each decomposer guild to decomposition. Sorry, I'm getting really distracted by the Kirby gift. I really like mechanism 3, so I'm really hoping that there's a difference in these carbon and nitrogen and phosphorus ratios between these burned and unburned plots. because this experiment will actually allow me a rare degree of mechanistic insight into fire effects on decomposition. So the statistical analysis here, just like in the first two mechanism experiments, will be another split plot ANOVA with two factors. So the big plot is still the burn treatment, and then the two factors here are meshed size for the bags, but then also litter origin. So did the litter come from a burned or an unburned plot? And like always, the dependent variable is once again the decomposition rate. And then my last chapter, which will hopefully be the last set of experiments that I do to look at these different mechanisms for how decomposition is altered by the reintroduction of fire, is going to focus exclusively on these earthworms because they are really everywhere at McCready, and you cannot step anywhere without seeing these invasive earthworms. So my last question that is all of my third chapter is, does the invasion of non-native earthworms alter decomposition rates via some direct effects like leaf litter consumption, or indirect effects such as influencing the relationships between decomposer guilds? And then can their invasion be controlled by fire? So I'm really focused on these earthworms because they're ecosystem engineers that can interact with all levels of the soil food web in any ecosystem. Lee Freelick has a great paper on how influential these worms are for ecosystems, especially after they invade. So I think it's really important to qualify the effects they may be having on how native decomposers are actually performing decomposition in the system. So just like I said earlier, in the literature, it's pretty well established that earthworms can increase the carbon dioxide given off by soils, while also increasing soil organic matter stabilization via their unique feeding behavior. But we don't know if these two effects are true for all ecosystem types. And we also don't know if the magnitude of either of these effects will be different if we couple the earthworm invasion with another ecosystem disturbance such as fire. And so there are multiple ways that fire can alter the earthworm population in burn sites. So first, burn sites have a more open canopy because they're killing off the trees. So this more open canopy allows for more sunlight to hit the forest floor, which elevates the soil temperature while reducing available soil moisture. And earthworms are really sensitive to changes in soil temperature and available soil moisture. They are really specific in where they like to live. Additionally, the lack of litter post-burn might force earthworms to migrate out of the burnt plots into unburned litter-rich plots. And then finally, the heat of the surface fires themselves, which at McCready, they do get pretty hot. The heat of the surface fires and the speed at which they actually travel across the landscape might kill the earthworms that dwell near or on the soil surface. So they might literally be dying because of these fires, and then their population might have to rebound afterwards. So what I'm going to do for chapter 3 is I'm going to try and exclude these earthworms and see what happens to carbon dioxide efflux. So how much carbon dioxide is given off by the soils. Here's my anti-worm agitprop on the right. So I only found one paper that tried to do earthworm exclusions on the field. So that's basically what I'm going to do. I'm going to copy their methods exactly. So I'm going to set up earthworm exclusion plots that are basically 36 centimeter cylinders made from metal hardware cloth. And I'm going to anchor them into the ground and then sell them into the soil each of my sites. Experimental plots will have the floors of these cylinders capped with fiberglass window screen that prevents earthworms from crawling back up into them. But the mesh at the bottom will also allow for water to flow freely to prevent my results from being confounded by changes in water availability, since we know that soil moisture is also a determinant of how fast decomposition rates and the CO2 efflux happen. And then to ensure that there are no earthworms within these experimental exclusions, I'm actually going to excavate the leaf litter and the soil down to the O horizon within each cylinder, and then remove all the earthworms by hand and then replace the soil and the leaf litter. And that's exactly what they did in the paper that I saw with the earthworm exclusions. So hopefully that will exclude all of the earthworms from each of these columns, and then they cannot get back into it because of this fiberglass cap at the bottom. So then for the control plots, I'm still going to excavate the soil and the leaf litter down to the O horizon, but I'm not going to remove any of the earthworms. I'm just going to put everything back. And then I'm going to allow all the cores to rest for a week or two to allow the soil and leaf litter to resettle. And then for this experiment, because I'm doing so much digging and installing these collars, I'm going to put them along their own transect in each plot away from where I did my invertebrate and earthworm extraction transects. And then I'm going to measure the carbon dioxide efflux from each of these plots across the growing season using a Lycor 8100 or a similar device. And basically what a Lycor does is it measures how much carbon dioxide is leaving the soil surface. And so I'm going to measure each earthworm exclusion plot twice a week for the duration of the summer growing season, because I want to get a lot of data from each of these little collars. And then the other thing that I want to do is I'm going to use Berlese funnels, which are right here on the right, to quantify the invertebrates at the soil surface and at the leaf litter layer, because I want to clarify whether any changes that I see in carbon dioxide efflux between our two treatments, whether it's because of this material loss of earthworms, so now the earthworms aren't around, or if it's because of some indirect effect of earthworm loss on the invertebrate community itself. So do I see different communities of invertebrates when I remove earthworms versus when I don't? Or is it just like the earthworms are gone and there's the same invertebrate community in these earthworm removals as there are in the controls? So that's why I'm going to do that. And I'll do that at the same time that I do the carbon dioxide measurements. So for the stats analyses here, it's pretty simple for the first part. So I'm going to do a t-test to ensure that there is some difference in the earthworms captured between control plots and experimental plots. And hopefully that will be, there are no earthworms in the experimental plots if I set up the collars right. But then I'm also going to use my field data to inform an SEM. So I'll make a meta model, and then the SEM will be parameterized using the data I've already collected from the experiments I did in chapter 1, and then the experiments I'll do in chapter 3. So here's what my meta model looks like. So for the simplest meta model, that's the stuff in orange, where earthworm presence is a yes or no. And then I'm predicting that earthworm presence will have some negative effect on the non-earthworm invertebrate abundance. But earthworm presence will also have a positive effect, so it'll increase the carbon dioxide efflux to a system. And then any change in the non-earthworm invertebrate abundance, so if there's increased invertebrate abundance in the absence of earthworms, that'll have a positive effect on CO2 efflux as well. So there'll also be more carbon dioxide coming off, depending on earthworm presence. And then while the first SEM that I just explained in orange might be informative on its own to show how earthworms are affecting this non-earthworm invertebrate community and affecting this carbon dioxide efflux, I also want to investigate how fire affects this system as well. So then I'm going to create a second SEM, which adds fire as a layer to the meta model, which you saw previously in gray. So fire is predicted to have a direct negative effect on both earthworm presence and non-earthworm invertebrate abundance. But then I also expect fire to have an indirect negative effect on carbon dioxide efflux, since fire is also eliminating the leaf litter that decomposers like earthworms and the other invertebrates would theoretically consume. So this added layer to the meta model would also be parameterized using my field measurements from chapter 1 and chapter 3, like burn treatment, earthworm abundance differences between the two burn treatments, and the non-worm invertebrate community differences between our two burn treatments. Ooh. So here's what that meta model would look like. And then for my proposed timeline, since I'm running short on time here, the chapter 2 experiments are going to be this fall for the decomposition experiments for mechanism 1. You've probably all noticed that fall has been a little weird here, and leaf abscission is kind of barely starting right now, which is super spooky. So I'm doing the decomposition experiments now, but I'm also doing leaf litter collection for the decomposition experiments for mechanisms 2 and 3. So thank you, Dr. Meisel, for letting me use your litter fall traps again. And then next fall or fall 2022 is when I'll do the decomposition experiments for mechanisms 2 and 3. And then the chapter 3 experiments, so my earthworm exclusions will happen in next summer. Oops. And now, are there any questions? There are 7 minutes left. I'm going to stop sharing if that's okay, so I can actually see people's faces. Hi. Many claps. Ooh, there's 25 people here. That's so scary. Cool. Are there any questions? You can just unmute yourself. Thanks, Serena. Sorry that I kept stuttering. I just get really nervous. No questions. Spooky. Thanks, Monty. Thanks, Blair. No questions. I'm spooked. Ooh, I have a question. What are we gonna do if we find jumping worms at McCready? Because I'm starting to get a little nervous.
Looks like Tim may have his hand up. Tim, do you have a question to ask?
What's up, Dad?
Tim always has his hand up because he'll always be one to ask a question. So while I was learning so much listening to this fabulous presentation, I was also, of course, doing some research on some birds that breed at McCready Reserve. And looking through the list that I was able to download and sort while you were speaking, I see 1, 2, 3, 4, 5, 6 that are pretty common and are ground nesters. So has there been any work done there? Not to add anything to your project, but has there been any work done there to look at potential effects of sort of the ground layer on these ground nesting birds that breed at the site?
I don't think so.
Yeah, I can jump in here. I think the answer to that is no. Although anecdotally, we've actually kicked turkeys off of their nests during our fire preparation.
That's terrifying.
Yeah.
I've done that once, where I almost stepped on the turkey.
Yeah. So it wasn't during the fire itself. It was during our fire preparation, but they've got to be there during the fire as well. So certainly probably some effects on any ground dwelling organism that can't get out of the way.
Yeah, not so much just during the burn itself, of course, but just what that means for structure, litter layer, and that kind of thing, and how that could affect them.
I wonder how well the ground nesting birds could actually nest in certain places at McCready, because in some places, like the rock layer is very close to the soil surface, and so there's also very little vegetation. So like all of McCready is generally extremely heterogeneous. But that's especially true once you get into like the different burn treatments. So what do you think would have a bigger effect? Would it be like vegetation mediated changes because of the burn treatments, or the heterogeneity of the soil at McCready for these ground dwelling birds? I don't know anything about birds. I know nothing. Birds aren't real.
Yeah, it's true. They are drones. We can put them wherever we like. Yeah, sometimes these ground nesting birds can be pretty flexible. So I've seen them in reclaimed strip mines and things like that, finding just a little patch of vegetation and it's enough to kind of tuck their nest away. But there are things like ovenbird is one of those, which is a warbler that nests on the ground. And for them, it's like leaf litter is where they hide their nests. So it doesn't even really need to be a lot of vegetation cover, certainly not live vegetation cover necessarily. But you got song sparrows, eastern towhees, ovenbirds, veeries, field sparrows, and blue-winged warblers as fairly common breeders there. So that's a nice little mix of different ground nesting birds.
Actually, now it makes sense. Some of the little birds that I always see on the forest floor, because I always think like, oh, they're just investigating me because I'm literally the lone human in these plots and I'm handling bugs.
No, I sent them.
Liz Anna Kozak asked, do birds break into traps like the raccoons? No. So the landscape staples are actually pretty big. I have a cup full of them at my desk, like a weirdo. And so they're actually pretty deep. Let me get it. So compared to my face, they're pretty deep. And so I actually put them very far into the ground. So there's not very much space actually between the soil surface and the roof of my cup. And so really only things with tiny little grabby hands like raccoons can be strong enough to actually grip it and like rip the roof off. Um, Whereas the little birds cannot get under that well. And I've never seen a big beak bird messing with my plots. So no, birds do not break into my traps like raccoons. I will say something I did not expect to happen as often as it did was a deer will step on my traps and just completely destroy them. I would think that the deer will be able to see it because they're bright red and they're giant soup bowl plates, but no. Usually 1 or 2 traps per collection cycle has been completely decimated by a deer just stomping it out. Blair Voss asked, was there enough human activity that there were concerns about people uprooting your sampling traps? If so, how did you mitigate that? The answer is I did not mitigate that. We set up some game cams to see like if there was actually people like yoinking my traps. But like some of my traps are very close to the public trail system, but I think it's more likely that it's just raccoons uprooting my traps and not people messing with it, or at least that's what I'm gonna keep telling myself. So yeah, I am always concerned about it, but I sample so much that hopefully like even if for 1 week or 2 weeks I don't get any data from a pitfall trap itself, there's still 7 other pitfall traps in the site and I'm going to be sampling from it so many more times throughout the growing season that it should be okay. I'm a little bit more worried about the earthworm exclusions since they're literally just going to look like PVC pipes sticking out of the ground. So I'm probably going to put up signs for that when I do it. But I think everyone has seen decomposition bags at this point, especially because of the light up studies. Those are really well publicized. Manny Jade asked, that picture of you in the mud is intense. How much time do you spend doing that for your work? Last year I had an undergrad with me, so 5 plots took all day, so 8 hours. But when I do it alone, it's 3 plots per 10-hour day because I have to carry so much material with me. So this summer was a lot for me carrying like all of my traps and then all of the mustard solution and then all of the samples. So that was rough, especially the swamp crossings because it's really easy to get lost. So yeah, it's a lot. It's a big time sink, but once you get all the data, you feel a lot better about it. Thank you, Lizanna. It's 3:01, so I'm gonna move on to the closed door portion of this. But thanks everyone for coming, especially all my friends from Twitter. Yeah, have a great day, everyone. Thanks for showing up.
Good job. And thanks, everybody.
Bye.
Great job, Toby.
Thanks, McKenna. Thanks, Nan.
So, yeah, well done. So for Toby's committee then, so I guess, why don't we take a break? I don't know, be back at 3:10, does that work?
Sure.
And we'll rejoin on the other Zoom link that I sent around.
Okie dokie.
All right, see you then.